Hi, my name's Cameron Dyer, and I'm going to talk to you about epistasis. So epistasis is defined as a gene that is masked or suppressed by the action of a gene at a different locus. So in other words, it's the process by which two different genes at two different loci have control over the resulting phenotype of the organism. This takes place by one gene masking the results of the other gene. But what does this mean? Let's look at an example. In honor of the upcoming July 4th holiday, we'll call this the America flower. So let's assume that coloration in this particular flower is controlled by two different genes. One gene that encodes for either red or blue color, which we'll call the A gene, and another gene that allows the petals to take up that color, which we'll call the B gene. So there are several patterns by which epistasis occurs. We'll discuss the single recessive epistasis pattern, where the presence of a homozygous recessive genotype in the gene that takes up color will mask the color coded for by the other gene. In the case of the America flower, if the B gene is homozygous recessive or displaying two small B alleles, this will result in a colorless or white flower, regardless of what the A gene codes for. Let's look at an example of a dihybrid cross and what offspring we can expect when recessive epistasis occurs. By crossing two parents who are heterozygous for both genes, we'll obtain the following breakdown of genotypes: 9 big A something, big B something, 3 little a, big B something, 3 big A something, little b, 1 little a, little b, or a 9 to 3 to 3 to 1 ratio. However, because the presence of two little b alleles results in a white flower, not a colored one, the resulting phenotypes will not follow that pattern. Instead, we'll get the following breakdown of phenotypes. 9 will be red, 3 will be blue, 3 big A something, little b will be white, and 1 little a, little b will be white. In other words, a 9 to 3 to 4 ratio. Now let's look at another cross, this time using one parent whose genotype is big A, big B, and the other whose genotype is little a, big B. So one is homozygous dominant B and the other is homozygous recessive A. We obtain the following breakdown of genotypes: 8 big A something, big B something, and 8 little a, big B something. This results in 8 red and 8 blue flowers in the offspring. There's no white flowers at all because there are no offspring with homozygous recessive B alleles. Therefore, the expression of red and blue colors is not inhibited, and the colors are taken up by the petals. But how is this different from what we learned before? Previously, when we looked at dihybrid crosses and compared the effects of two different genes on an offspring's phenotype, we were looking at two different genes. For example, one that coded for petal color and one that coded for plant height. Epistasis differs from that because both genes that we're looking at code for the same characteristic. In this way, epistasis is similar to patterns we observe with dominant and recessive alleles, where the presence of one dominant allele masks the trait coded for by the recessive allele.